So this first lecture is to give you a basic understanding of what a virus is and what it is not. So the word virus comes from the Latin word for poison or venomous substance or slimy liquid. I'm not exactly sure how all of these make the word virus, but one of the reasons we'll talk about in a minute that it was thought to be a slimy liquid is viruses were first discovered in the sap of plants as something that was contagious or infectious from plant to plant. So this gives you a basic idea of what a virus is. I'm just trying to peek. Virus is a capsid, which is a protein shell, and genetic information, which we'll talk about could be DNA or RNA. Some viruses also have an envelope, which is a phospholipid bilayer, which is taken from the host cell. So this is called a naked virus without an envelope, and this is called an enveloped virus with an envelope. We'll talk a little bit more about these differences and some examples of them. But basically all viruses have a protein shell, which we call a capsid, and some kind of genetic information inside it. A virus is not a cell. Okay. A virus is an obligate intracellular parasite, which we'll talk about in a minute. Oh, no, I'm good. Okay, thank you. Sorry, just got a knock on my door. Okay, so let's go on. Size of a virus. Viruses are really small. Okay, so yeast cell, which is a eukaryotic cell, is shown in blue. Bacterial cells on the top. One of the bigger viruses that we knew of for a long time, the pox virus, herpes virus, you can see their nanometer size. Polio virus right here, super small, 30 nanometers in size. So viruses, you cannot see them with a regular microscope. So you have to visualize them with an electron microscope, which is another reason we didn't really know about viruses for a long time, because we didn't have the ability to see them. We have recently discovered some large viruses. Okay, so the Mimi, the Mama, and the megavirus. So here is HIV, which was back here, number 4. Okay. And you can see these are much bigger, 400 nanometers in size. They're shown over here. They've got really unusual structures with these protein fibers. Very big, first isolated in amoebas. So they're actually close to the size, or about a fifth of the size of an E. coli bacterium. Some can be seen actually with a light microscope. They're so big. So I mentioned viruses are obligate intracellular parasites. They're not cells. This means that they must replicate inside a host. And they're parasites because they end up, for the most part, destroying the host cell. Now we'll talk about some different types of infections, but in general, they don't do anything good for the cell. They're there to take over the cell machinery and build more viruses and get out. There is a debate whether people classify viruses or living or dead. I personally go by the standard that all life is based on cells. Viruses are not cells, therefore they are not alive. They cannot do anything metabolically outside of a cell. But if you want to debate that, we can debate that at another time. Couple important concepts about viruses is specificity. Okay, viruses only infect certain cell types, and that's mainly due to host cell receptors. Okay. So in general biology, cell biology, you learned about receptors being important for cell signaling and cell communication. Viruses take advantage of these naturally occurring receptors, and they interact with them on a host cell and kind of trick the cell into letting them in. If a cell does not have the specific receptor, the virus cannot infect it. So the host cell receptor is that first line of specificity. There are certain host ranges, so types of hosts that a virus can infect. Tissue tropism, which means which cells specifically the virus can infect inside a host organism. But these are all components of the specificity. Also, a host might have the cell receptor, but an infection might not happen if the host cell doesn't express the right factors. And I know factors is a really general term, and we're just going to say things like proteins. So for instance, polio can infect some cells, but they don't have the right host factors needed for polio translation, and so no viral proteins are made, and so polio has kind of an abortive infection in those cells. So we'll talk about this more, but specificity is a really important concept. All right, so you know a little bit about viruses. They're basically genetic material in a protein coat, capsid. How were they discovered? Well, most pathogens were characterized using Koch's postulates. So the first step is the microorganism must be found in the diseased host. Okay. And you must be able to see it. So that's what found means, is they were using a microscope. So they could take a mouse that had died from an infection, they could isolate and see the microorganism. Okay. The problem with viruses, they're too small for the types of microscopes we had back then. Remember I said you have to use an electron microscope to actually be able to see the viruses. the next part of Koch's postulates is microorganism, whatever's causing the disease, must be isolated and grown in pure culture. So they knew lots of ways to grow bacteria, fungi, things like that. You could see protozoa under the microscope. But the problem with viruses is viruses require a host cell to reproduce. So you can't grow a virus without what we call tissue culture, without a cell. So they weren't able to satisfy the second part of Koch's postulate. The third was that the microorganism must cause disease when put into a healthy organism This would definitely work with viruses if you could grow them. And the fourth part is the microorganism, the same one that you got in step 1, must be re-isolated from this second host. And just like we said, up here, it's very hard to, at that time, isolate viruses, grow them up, see them under the microscope. So there was lots of ideas about what a virus was. People thought it might be just something in the air. There's an old term called miasma, that just the air is infectious and causing issues. Remember, again, if we couldn't see it, we couldn't necessarily understand it, and we didn't have really strong tools to visualize. But some people were working on plants that were showing disease. So a diseased tobacco leaf will have lots of spots, cell death, necrosis, browning. And so what these scientists did was they took these diseased tobacco leaves and they crushed them up and they made it into this slimy liquid, which is called sap from a plant. And then they put it through a filter. Okay. And the filter would, let me write this over here. The filter was big enough to retain bacteria. But things that were smaller could go through. Okay, so this liquid had stuff that was smaller than a bacteria, so filterable. This term up here, contagious living fluid, is talking about this liquid down here. They didn't know it was particles at the time. They just again thought, okay, instead of air, now it's a contagious liquid. And what they did was they would take the cell debris, the stuff up here, and rub it on the plant, and the leaf was healthy. So the disease-causing agent wasn't large, right? It wasn't retained in the filter. But when you took the cell filtrate, the liquid, and rubbed it on the on the leaf died. So they realized that the infectious agent was in the liquid. or the filtrate. This was kind of as far as they got for quite a long time until we could start visualizing and learning that viruses were in fact these particles that were floating around in that filtrate. So we're going to jump from the late 1800s to the mid 1900s, where Hershey and Chase showed that viruses had genetic information that was DNA, not protein. So remember, in the 50s is when people are figuring out the structure of DNA, and that DNA is the genetic material. And so what Hershey and Chase did is they had these bacteriophage. So bacteriophage are viruses that infect bacteria. And they could use radioactive, radioactive atoms. So they use something called 35S, which is sulfur, and sulfur is found in proteins, not DNA. They could also use 32P, which is phosphorus, and that labeled the DNA. Now remember, I said viruses can have DNA or RNA as their genetic information. They happen to be working with a DNA virus. And so what happened is they would have two sets of these bacteriophage, one with radioactive proteins, and this is horrible that they say capsule. I need to fix that. And one with radioactive DNA. And so they allowed these viruses to infect bacteria. And then, this was my favorite part, they used a blender, like your Vitamix at home, and that was to remove any viruses that were stuck on the outside. So now they just wanted to look at the bacterial cells. So they did centrifugation, which is spinning things around really fast. Heavy things like bacteria cells go to the bottom. Light things, everything in the liquid, stays up top. Right? So you would have a little test tube with your bacteria down here, and your liquid is going to have the virus up top. And what's unique about bacteriophage is they actually inject their genetic material into the cell, and the capsid stays on the outside. So what they found is when they had radioactive protein, the infected cells were not radioactive. All the radioactivity was in the liquid. So that means the proteins were staying with the virus and they were not getting injected into the cell. That suggested that protein was not the genetic material. To complement that, they did the second experiment where they had radioactive DNA. And when they looked at the bacterial cells, that's where the radioactivity was. So the virus was injecting its genetic material into the bacteria, which was DNA. So the conclusion was that viruses had DNA as genetic material, just like people were realizing with cells. And this page that it keeps flipping to, because technology does not like me, here is an actual electron micrograph, and these really are these two T2 phage, and they're so cool. They look like these little lunar landers. This is your cell in yellow, and you can see they're injecting their DNA right into the cell. And then the capsids just fall off. So where did viruses come from? The real answer is we don't know. there's kind of two big schools of thought. One, that these were vagrant genes. Depending on how you count, 5 to 8% of the human genome is retrovirus sequence. Yes, like HIV is a retrovirus. Now these are dead in the sense that they cannot replicate, they can't make virus. They're called endogenous retroviruses. But because they look like viruses, the idea is maybe these were genes that kind of just moved around. The second idea is that they're really degenerate cells, so that they were once cells that lost their ability to reproduce on their own. And they just kept the genetic information and became this virus. So kind of like degenerating from a full cell just to genetic material in a protein coat. We really don't know where the first virus came from. We do know that every cell type that we have looked at has some kind of virus that can infect it. We find viruses in every type of ecosystem. So yes, amoebas can be infected by viruses, plants can be infected, bacteria can be infected. Obviously, humans and other animals can be infected. Every type of organism that researchers have looked at have found that viruses, some type of virus can infect them. The evolution of the virus genomes is also an interesting question. The kind of current, oh no. I don't know if I'm still recording. Oh, I think I hit a button. The current idea is that genetic material really started as RNA. And RNA has some cool properties about it. RNA can be a ribozyme. This RNA is not translated, it's not made into protein, and ribozymes are like the word enzyme, but made up of RNA instead of protein. So we think what happened is these nucleotides started joining together, they got functions, they could cut things, they could self-replicate, and likely RNA viruses were the first type of virus there. Retroviruses have RNA genomes that are copied to DNA, so those were kind of the next or the middleman, and then DNA viruses evolved. And that's the same idea as what we think of what that happened with cells, is that RNA was this first genetic material, maybe not of a cell, but that DNA evolved as a more stable type of genetic material. So that's what our cells have now. This graph down here is just trying to show you that there's a relationship between error rate and genome size. So when you learned about cells and DNA replication, you learned that DNA polymerase has some proofreading capabilities, and you may or may not have learned that we have tons of DNA repair mechanisms in our cells to keep this DNA, the genetic material of the cells, as pristine as possible. Well, there are RNA viruses that have a very high mutation rate. viruses like the flu, influenza. The reason you have to get a shot every year for influenza is because it constantly changes. So RNA viruses don't have proofreading, and so they have a high mutation rate, and they keep their genomes very small. Okay. Keep the basics. As viruses evolve to get double-stranded DNA genomes, they had a lower mutation rate, and they were able to evolve kind of larger genomes. And then you can see cellular genomes here, low mutation rate, and larger genomes. So as you get larger, you need to become more stable, lower genome rate. All right, the last slide I want to talk about is to kind of end this, why we're talking about genomes. One, all viruses have them, and I want you to see that we'll be talking about the virus classification, the Baltimore system, which is based on the type of genetic material a virus has. So there's 7 classes. You can see it around here. And viruses can have double-stranded DNA. They can have single-stranded DNA. They can have double-stranded RNA. They can have single-stranded RNA. They can have positive sense, which is like mRNA. They can have negative sense, which is the opposite strand. And the retroviruses can go from positive stranded RNA to eventually double-stranded DNA. The classification system is divided up as to how the viruses get to messenger RNA. And the reason we focus on messenger RNA is this is how the virus proteins are expressed, which then starts the whole replication cycle for the virus. So I'm going to stop there. The next talk we'll talk about what viruses look like, a little bit more in classification, and we will get into what a virus is not.